Hi, everybody. Welcome to It's That Easy, 10-minute answers to your drug discovery challenges with me, your host, Jeremy Jones. I'm principal scientist of AI-driven drug design and Simulations Plus. And as an academic turned early drug discovery scientist, I like helping others find faster, more efficient ways to tackle their drug discovery and development challenges. As always, we're keeping it cash, and I promise to deliver the message in 10 minutes or less, or your next episode's free. Today we're going to talk about building some great QSAR models. QSAR, I love that acronym. It sounds so space age. The quantitative structure-activity relationship model. It's an incredibly useful tool for early drug development, and it helps you to figure out, what features of chemicals are important for a particular activity just from the structure of the molecule. We often apply this to activity at a specific drug target, but really the same principle can be extended to any property, permeability, solubility, whatever. But let's think about a typical scenario. You screen a library of, thousands and thousands of compounds against your favorite assay. Let's say the assay is one of my classic favorites. You're looking for inhibition of a particular target using a luciferase reporter assay. So, you got your, protein that's, when it's activated, it's going to bind a response element, and that will turn on, the activity of the luciferase. So you see the luminescence. By the way, you can obviously tell that, graphic design is my passion. If you're looking for inhibitors, maybe you're screening small molecules, you're looking for something that blocks that entire activity, right? And so your readout is in a plate format where you have a bunch of wells that are lit up with luciferase activity, and then a handful that are dimmer, right? That might be possible hits. And so you'll pick out, a couple hundred or so, to follow up on, and you'll go back and rescreen them in the assay, maybe 1 or 10 micromolar, to see if any show any follow-up activity that are real hits. And, about half of them do or something. Great. You're going to go back and take those, and you're going to screen those more closely with a larger dose curve to get a more accurate inhibition constant 50, your IC50 value. Fantastic. Now you've got yourself a good starting data set. Well, in some cases, you'll want to start with this. If you're a little further along the development pathway, you might have a very specific lead series scaffold, and you'll want to keep your QSAR model very focused on that specific scaffold. In this case, we're talking about, a little bit earlier. We want to try to cover as large of a chemical space as possible with our QSAR model. And so we've got a good starting data set in our lab, right? But you might know of other folks in academia or industry that are doing similar work and have done some screening or identified some molecules that might be also inhibitors. So we look in the data and for the data in the literature. And I just want to point out that you really do have to make sure it's compatible. Like, they ran an assay very similar to yours. There's always lab-to-lab variability, but hopefully they ran some standards if there's any known inhibitors out there. And the IC50s looked very similar to what you saw in yours because you obviously ran those controls standards as well, right? And if they are, you feel comfortable combining those data sets, well, then you've increased your chemical space and the size of your data. If you want to go, I'll make a little plug for myself here. I go into depth about data curation and when it's okay to combine data and things like that. So feel free to check out that blog if you're really bored or can't fall asleep. Click on that link below. All right. So now let's say we've got, maybe 200 compounds or so with yes or no data, right? We rescreened it at 10 micromolar. In 1 micromolar, we saw some didn't have activity. So we don't have an IC50. But we do know that we're confident calling them inactive. And then we have, a little bit fewer data with IC50 data, fewer points with IC50 data. The reason we might consider making 2 different models, one, a classification model, like is it yes or no, predicting yes or no, whether it's an inhibitor or not, and 2, a regression model where we'll actually predict the IC50, that we want to cover as much chemical space as possible and, theoretically, we can use all of the compounds for which we have IC50 data. Those are active, and we'll set an arbitrary cutoff. It can be whatever you want, but here we'll say 10 micromolar. We have even more compounds that we know are inactive. And so then we can build both these models, and then we can set it up so that the regression model will only be employed if a compound you're evaluating first passes that classification model. Again, it's a great way to use as much chemical space as possible and still get our very reliable IC50 predictions. Okay, so you have the data. Now what? Well, there are any number of software options you could use to build your QSAR models, many of which are freely available from places like GitHub. Absolutely worth checking out some and reading about the various advantages and disadvantages. I work for Simulations Plus, so I use our fancy ADMET modeler software. It's free for academics, by the way. But some of the things that I want to point out quickly before we get into it about QSAR model building are applicable really to any software. Things like making sure the structures are correct. Often when you're going through the data, you realize that the structures aren't actually what they're supposed to be, so sometimes you have to check on those. You want to make sure they're the most favored tautomers, things like that. You want to use a high quality and a wide range of descriptors. So if you're familiar with this process, descriptors are ways to identify different features of your chemical, and they come in different flavors from, just 2D descriptors, some are 3D descriptors. You probably want to make sure that you're not just using the standard ones off maybe already kits, which aren't bad, but look into a little bit about what descriptors your models are using. You want to protect against overtraining. Some models look too good to be true. They are. You want to make sure you have very unbiased test sets. You pull out some of the data from your data sets and set them aside for test sets. And you really want to look for the relevant performance statistics. And we'll talk a little bit about these as I show you how to build some of these models. But if you want an in-depth read about this, here's another blog that I wrote. Sorry to keep doing it. I promise that's the last one. But that link's below too if you really want to go more into depth about what to look for in these models. So for instance, here's a data set that I was looking at for some PPAR compounds where I have different identifiers, some from my old lab, some from the literature, and I just have the identifier, the smiles, and then I changed the IC50s to the negative log of the IC50s, PIC50s, to put them more in a closer range that just facilitates model building, something I would suggest for you as well. I've loaded that data into our ADMET predictor. Here you can see that same graphic you saw in the PowerPoint. Here's a categorical one that I've already loaded into our ADMET predictor software. Again, free for academics. And then we just go up to our fancy modeler tool here, and it will automatically recognize the different user input dependent variables. Here I just have listed it whether it's active or not, 0 for inactive, 1 for active. And it recognizes that. The first screen this brings up, this shows you all the different descriptors we have for training. I'm going to run through this a little bit quickly. If you are interested in any of the details, feel free to contact me or look on the website. We'll go into this more in detail for sure in some of our other webinars. So we're looking for a classification. We'll get to the regression a little bit, but we offered various different techniques of model building. In the classification, you can use a support vector machine or an artificial neural network. I'm going to use the artificial neural network. You can dictate how that set-aside test sample will be. We have a couple different options here. We'll just use some random stratified sampling. We'll set aside 25% of the data set. And also you can dictate how the descriptors are selected to go into the model building. This genetic algorithm by cell is a very powerful technique of picking the best descriptors. I'll go on to the next one. And this just it says what range of different number of inputs or descriptors are going to go into it and how many different neurons. We are going to just, for the sake of time, really limit this to, I don't know, we'll just make a couple here. But really, as you'll see, so I'll hit build. We'll have a name for it. It asks to which class represents the negative category, and that's 0. So it's going to start building different neural networks. And then it will tell you, based on our different metrics, performance metrics, which ones it thinks are the best. And so you can look at the sensitivity, the specificity, or this Uden value, which kind of takes both into account. And when you click on one of these, it will bring up this cool little chart. You can see this is for 8 different descriptors or inputs with 4 neurons. It'll tell you the false positives and false negatives. Real quickly, I want to show you the regression model too, and then some examples of the ones we built. So the regression model, very much the same. We'll go up to Modeler. We'll pick our IC50. That's fine. We'll just skip ahead again. Again, we'll do our neural network. We'll just do the default for the test selection. We'll autofill here. We'll just do 1 to 3 and 9 to 20, just real quick so you can see what some of these look like. And it's building these models. And of course, you would go back and try this a couple different times to really hone in on what I think of is the topography of what might be the right number of neurons and the right number of inputs. But here you'll get a model that looks like this. Not so good. We didn't do too much. But thanks to the magic of me having prepared this in advance, I'm going to go back to this and then show you these sweet models that I had built previously. And so just looking at some of the metrics here, here's our classification model. You can see a really low false positive rate, really low false negative rate. The blue points are those in the training set, the red in the test set. You can see nice and spread out, high sensitivity and specificity in both the training and test sets. This is a nice classification model. And as I said, now, if we were predicting inhibitor, we would make it past this model before it even saw this one. And you can see some of the statistics for our regression model now. This one's built on 24 descriptors and 3 neurons. A nice wide range here of this is the observed versus predicted. And you can see a low root mean squared error, a high Q squared value, which is similar to the R squared. A lot of different performance metrics here. With those sweet models, I just wanted to sort of wrap up here. Those were pretty good models with small to medium-sized data set. The more data you have of high quality, the better you can expect your model performance to be. Now, these models are ready to be implemented to predict the activity of the other molecules might be designed in your fancy computer programs or whatnot to help you decide what molecules to make and test next. I'll discuss things like that and much more in other episodes of It's That Easy. Thanks for listening.